I mean, I fancy other people, but this one's for real. Mark and I have worked together now, on and off, for about 17 years.
That sounds too long.
When you look at me and my beautiful, freshly coloured hair.
Versus mine.
We like to joke around. Stop it off blood. Some embarrassment. But anyway, so I actually was one of the lucky people to examine Mark's thesis.
Oh, yes.
Thesis,
Yes.
Just for everyone's sake.
Yes.
Reagent is spelled with an
Yeah,
A.
she remembers. And so do I. Never made the same mistake again.
There we go, see? So Mark has been working for QT, did his studies at QT, went over and worked at Varda for a while, did a PhD there mostly, came back to QT, is a group leader now, leading his own group, but as part of the group that's over at IBI, and works alongside people like Aaron, who's coming up, and Derek, who you've already had. He does some really cool stuff with DNA repair, and I think your talk today is on breast cancer stuff.
Yes, largely, yeah. Yep. In fact, all of it.
Enough about me.
Yeah.
Take it away.
Yeah, nice. Thanks very much, Sally.
I forgot. We have a plan.
Oh, yes.
Our plan is, because we felt that asking questions at the end actually worked well, except for the fact that I went across the room and people were sitting there great with their hand up. So we've got numbers. So what we're going to do is let Mark talk, and when he's finished, everyone who wants to ask a question will be assigned a random number, no 12, that are handed out, 13.
Oh, wow.
That will be handed out by me and Mark randomly, and then we'll just go through and do 1, 2, 3, right? We thought that'd be fun. Anyway.
Just to mix it up.
Yeah.
Why not? So, yeah, as Sal said, I'm going to try and rip through this in about 20 minutes to give us bulk question time at the back end. But if you're feeling super keen, I'm going to completely ignore you. No, all good. Yeah, so as Sally said, I really appreciate the chance to come and have a chat again today. Have lots of fun. I think it's the question time I really enjoy. So hopefully you enjoy it too. So what I'm going to do today is tell you a little bit about some work that we've got currently under review for publication in a journal called eBiomedicine. Sounds super amazing. It's not bad, but I'm hoping it gets up. It would be lovely if the reviewers accept it. And that's all about identifying this gene network and some specific hubs within that gene network that regulate or influence that gene network and how that is related to chemotherapy response. And today I'm talking about breast cancers. But where a lot of our work really sits, this central grey box is what sort of drives me. So current approaches that we have in the clinic at the moment really do, they're effective for some, but not for all. But they're not adequate for everyone, okay? So they're failing people that are living with the disease. So we can do better. So what we have at the moment is a variety of traditional and personalised approaches to treat solid cancers. You've probably heard about them all. My work tends to focus predominantly on chemotherapy and targeted therapies, largely in lung cancer. So that's why I've popped that little Lung Foundation logo down the bottom corner. So just hands up, who's got lungs? I'm not seeing all the hands here. I'm getting a bit worried. That's all of you. So because we've all got lungs, we all could get lung cancer, all right? There's a lot of stigma about lung cancer when it comes to smoking. When you hear about something diagnosed with lung cancer, they may say, you might think, or others, you might hear others say, did you smoke? Don't do that, because more often than not, never smokers, and if you're female, you're at greater risk of developing lung cancer, okay? So smoking isn't the only cause. It's a risk factor, and it's causal. It's multifactorial, okay? So there's a lot of stigma about lung cancer. But that's about all you're going to hear about lung cancer today, maybe just a wee bit more. But how do we go about identifying who it is that may be a responder to chemotherapy or a therapy, and then who is it, and how may we better treat those that perhaps aren't a candidate for chemotherapy? Well, so what we're doing in our lab is using a variety of approaches. So I've put the magnificent triangle in there of multiomics combined with molecular modelling using computers to sort of understand the proteins that we're studying and the enzymes. And then at the base of all of that is experimental validation. So we'd love to get our hands dirty with gloves on and do our experiments. What we're trying to do is identify druggable targets, druggable strategies for these targets, unique combinations of these new novel agents alongside the standard of care, ultimately with the goal of delaying at the very worst, but ideally preventing therapy resistance. How do we go and do that? Well, one of two approaches. One, we're going to need biomarkers. Okay, we need to screen people upfront, ideally in a minimally invasive approach, so a blood sample or even just a body scan. How can we use those technologies to better work out and predict how someone's tumour may respond to treatment? So there's some biomarkers or the molecular features of that particular tumour, as well as developing those novel anti-cancer drugs. But in order to do that, we need to have a deeper understanding of the biology of disease. So I'll put lung cancer in there, but I'm going to talk more globally about solid cancers rather. So discovery science is really, really important, and I really love it. So get amongst it. So how do we do that? Well, we can't do it alone. Science is a team game. You've got to work with folks. You've got to collaborate. I'm a big fan of that. I'm going to talk about collaboration on the next slide. That discovery science I spoke about. Yes, we have to go about, get out there, write some grants, try and just make some dosh, secure some funding, all with the end goal of improving health outcomes. So there's a couple of granting agencies I've been able to get into and score some cash from. Right. So as I said, we're going to be talking about my breast cancer work today and the manuscript we've got under review. And what we did is we first collaborated with this lady, Jodie Saunders, a couple of years ago now. So this has been a couple of years in the making. Takes a while to do our work. So Jodie published a paper back in 2022, many years ago, great vintage, using this approach here, weighted gene co-expression network analysis. So that's basically just systems biology, okay? So we're taking on board a whole wealth of information. How do we put it all together, use some high-powered fluting computational power to look at networks within a breadth of tumours. So in this case, we've taken about 1,000 breast cancers. We've sequenced them, so not we, but we're using publicly available data as well as combined with some of what Jodie has done. Taking all that data, forcing it in to look at the connections between all the genes, how co-expressed they may be, and then how connected those genes may be to identify larger modules, programs that are functioning within a whole variety of tumours. Okay, so really using a whole lot of samples gives us a whole lot of power. And this is a sort of plot that we can come up with. So what we can do is we can stratify all the different breast cancers. From the far left, we've got basal, and towards the far right, we've got this normal-like. And in the middle, we've got HER2, we've got luminal A, luminal B. Essentially, breast cancers, just to be rather crude about it, can be measured by what it has and what it doesn't have. Okay, so we've got hormone receptors like estrogen, progesterone, as well as HER2 receptor and RTK, which Sally knows a thing or two about. So we can say the luminal A, luminal B, they're very hormone driven. So if we look at all the different modules that might be functioning within these breast cancers, you can see on this far left, and we denote them, give them a color. In these luminal A, luminal B, we're seeing this underlying brown. So what this brown is this hormone receptor signaling. So we can say these are hormone positive breast cancers. But what really interests Jodie and I is those tumours on the far left, these guys here, okay? And what we see here is this mix of green and blue. Now, I love these colours, but not because I love these colours. I actually am very, very interested in the biology that underpins mitotic instability and multipotency. And what we find here, these basal breast cancers, these are termed triple negative largely. So they don't have any hormone receptor, they don't have any HER2. And these cancers are considered the most aggressive of breast cancers. So we were very interested, given that there's this high proportion of green and blue in these cancers, we wanted to look at the connection between these two modules, because we really sort of hypothesized that there's a lot of stress and tension between these two modules. Tumours are trying to divide, but also stay really plastic and able to respond. So there's a lot of tension between those two. So we really were keen on identifying key hub genes and also genes that function at the interface between these two modules. And hey, presto, this is what we got. We just chucked it into PowerPoint and said, give me something. No, that's not what we did. So what we've done is we've performed some topological clustering, which is to get, like I mentioned before, about that co-expression and connectivity. So you can see all these links between all of these genes. And if you look a bit closely, you may not have as much love for this as what I do. But there are a lot of my favorite genes in this mix that really do control mitosis, so cell division, and the timely function of that mitotic process. So we've used this force-directed visualization approach to look at all of these different connections. And as I mentioned before, we want to look at those key genes, that interface in the center, but also at the interface between two. And what really did pop out is actually, would you believe it, one of my favorite genes, the CDCA3, cell division cycle associated protein 3. I'm just going to call it CDCA3 because that's a bit easier. But it has a lot of connections and co-expression and connectivity with a lot of these genes, as well as these green module hub genes, which is really, really cool. So to sort of look at how influential CDCA3 may be, we performed some bit of mathematical wizardry. What we did is we forced the upregulation, so the increased expression of CDCA3, to look at how stable this module is, as well as taking it away. So you can see by taking it away, 2 standard deviations of expression didn't really impact the stability of this particular network. That's what we're measuring here. But if we add more CDCA3 into the system, the module has all sorts of dramas. So what this tells us is that CDCA3 is quite stable for one, and then 2, it's really, really influential within these modules. So I really want to know a little bit more about what CDCA3 may be doing. But what do we know about CDCA3 at the moment? Well, CDCA3 is known to regulate the entry into mitosis, so the progression from the G2 phase in the cell cycle into mitosis. And it's reported to do so by controlling the expression, not so much expression, but the levels of this particular protein here, WEE1. WEE1 is an inhibitory tyrosine kinase. It phosphorylates CDK1. If anyone's heard of CDK1 before, yes. All right. CDK1, cyclin-dependent kinase 1. It's the master regulator of G2 and mitosis in combination with its cyclin B1. If this guy phosphorylates CDK1 not be working, so you're not getting mitotic progression. Take it away, like CDCA3 does, you can progress into mitosis. We've been able to publish a couple of years ago that CDCA3 is associated with poor prognostic lung cancers, and its levels are markedly elevated in lung cancer. The other really, really fascinating thing is this is the AlphaFold. You've heard about AlphaFold, right? Yeah, you have. This is the predicted structure for CDCA3. And you can see this is really intricate. There's lots of secondary structure going on here, right? Negative. This thing is a sloppy, dirty protein, just flops around. It's got a lot of unstructured regions, and that's made it really annoying to study. But what we have worked out, and I'm not going to talk about it today, is that upon binding to certain proteins, it does form secondary structure, which I think is pretty cool, right? So it tells a little bit more about the function. But again, I'm not really going into too much detail on that today. So that's CDCA3. What we know about it, we've published it in lung cancer. So we want to look a little bit more into the breast cancers. So what we've done is perform some immunohistochemistry on tissue microarrays. Everyone heard those words before? Yep. So we've stained for CDCA3 protein in a whole couple of hundred breast tumors, actually. And with the help of a pathologist, we've been able to score the expression levels for CDCA3 down from none through to more than some. And then we've been able to take those scores and associate them with clinico-pathological data. So in this case, we're looking at survival. Kaplan-Meier curves, people have seen Kaplan-Meier curves before? Yes. Woo. Survival over time, and the percentage of survival. And really what we found is that those tumours that had some high CDCA3 had a better outcome versus those tumours that had a little bit less. So this tells us that the expression levels for CDCA3 are highly prognostic, and there may be something more worthwhile to know about CDCA3. So why is it that those tumours, those high, have better survival? Let's ask the question. Let's look into it. So we, given our interest in chemotherapy response, what we've done is we've first gone back to our bioinformatics, looking at the CDCA3 transcript expression. So we got, as well as the module that CDCA3 functions in. And so we got low through to high module function, low through to high CDCA3 function. And we've correlated that with this PPSC. Everyone's heard of PPSC before? No, I don't expect you to. It's a multi-gene signature called the pharmacogenomic predictor of sensitivity to chemotherapy. So it's been published a couple of years ago. This genome's molecular signature is associated with tumoral response to chemo. So low PPSC, less responsive. High PPSC, more responsive to chemo. What we see here is that there's quite a positive correlation between the module activity as well as CDCA3 expression. So CDCA3 high, more responsive to chemo. That may help us understand why these CDCA3 high have better survival. So not only did we look at that against the PPSC, we've actually looked in clinical trial data. So what we've done here is, in this case, we've taken the module and looked at its expression in tumours that had a PCR, pathological complete response, so tumours that really responded to chemo or the treatment, versus residual disease that's not responsive. So what we can see here is that in these green, high module activity associates with tumours that have a complete response to treatment. So what we see here, and these are the two that I'm really going to focus on a little bit later, epirubicin and this TA or TEC. These are DNA damaging agents or antimitotic agents that serve to control the activity of an enzyme called topoisomerase 2. Topo2, which I'm going to talk about a little bit later if I get time, controls the torsion of your chromatin. Okay, it's there to unwind your DNA, which gets all wound up in a whole bunch of different processes. If that gets all tightened up, you don't like it too much, it's going to snap. So you need to control the torsion of your chromatin for a variety of reasons. As you go through DNA replication, as you're opening up the fork, if you've got too much torsion, again, like I said, it's going to snap. So we've taken all that clinical data, jumping back into the lab now. Everyone's seen a Western blot before? All right, I'm still loving my Westerns. They're pretty fantastic. We've taken a panel of breast cancer cell lines, taken their lysates, performed a Western blot for CDCA3, and these cell lines are from the variety of types of breast cancer that I spoke about on that very first slide. We've got a bit of hormone positive breast cancers and these triple negative on the far right. And what we found is much like what we saw in our immunohistochemistry data, that CDCA3 expression is highest in these triple negative breast cancers versus the hormone positive. And we've done some densitometry just to make sure our eyes aren't deceiving us. We've then taken all these cell lines and looked at their basal response to chemotherapy, so performed a dose response curve, a concentration curve. So we treated them with low through to high concentrations of this doxorubicin, which is the drug I was just talking to you about, okay, controlling topoisomerase 2. And we've looked at how live, or the viability of the cells. And naturally, you'll see the cells dying off over time because more drug, cells don't like it too much, right? So we can measure the potency of this drug by taking the IC50 value. So that's the concentration of drug where you see 50% of the cells hanging around. So we've plotted these IC50 values based on low potency, so high concentration of drug, versus the super potent. These only need a little bit of drug to kill off 50% of the cells. And again, what we see here is much like what we saw before in our clinical trial data. CDCA3 high cell lines are more sensitive to doxorubicin. Now, what do you reckon we should do next? Do you reckon we should do some more experiments? Who's up for that? Yeah, I am. I don't believe you. Totally. That's great. If I was to do more experiments, would you like to test this out by taking CDCA3 away? Hands up for taking it away? Yeah, that's all right. Hands up for putting more back in. Yeah. Look, both are good. I'm not going to show you the taking away. I'm going to show you what we did to add more. If, based on these data, what would your hypothesis be? By adding more CDCA3 in, what do you reckon the outcome might be? What do you hypothesize? Increased sensitivity? Yeah? Well, would you believe it? You're absolutely incorrect. No, I'm just wrong. That's what we saw. By overexpressing CDCA3, we increase the sensitivity to both doxorubicin and cisplatin, two different chemotherapeutic agents that have slightly different mechanisms of action, but ultimately they're inducing DNA damage. Your beauty. So we want to know a little bit more about why on earth this is going on. So again, we took these cells where we've ectopically or increased CDCA3 expression. What we found is that those cells, when we overexpress CDCA3, have an increased mitotic index. All that's telling us is you, in overexpressing CDCA3, got more cells hanging out in mitosis. That might make a little bit of sense, right? Because it's playing a role in progression through to mitosis, which is fine, but the rest of the data I found a little bit fascinating. So if we're increasing the mitotic index, we then looked at proliferation. When we overexpress CDCA3 in this grey line, we're actually slowing down proliferation. So we're not speeding it up by speeding up entry into mitosis, which we may be. Maybe next year I can tell you about it. But I think what we've got going on is we've got cells piling up in mitosis, so they can't progress and exit out of mitosis. So we want to look at the cells. Let's do some microscopy. Seeing is believing, as they say. We want to look at the nuclei, what's going on there. We can tell by the nucleus when we stain for the DNA what stage of the cell cycle they may be in, as well as what stage of mitosis they're in. You'll remember that back from first year, right? You can look at the different phases of mitosis. Well, someone else may have. I hope you learned that. Anyway, I love doing that. By just looking at cells in the microscope, staying with DNA, you can tell what stage of mitosis they are, right? And in doing so, you can tell what stage they may have failed in mitosis if there's some mitotic issues. In this case, we've got quite a few issues. We've got these micronuclei, right? Micronuclei, what's up with that? You've got these tiny little nuclei, right? And that comes about when a cell actually exits mitosis with dramas. So they've got all this extra DNA that what am I going to do with it? Oh, I'll just shunt it over there. Essentially, that's a micronuclei. So they've exited mitosis, they've gone through into G1, and then they may have come back through to mitosis, and that's where they're having issues. So that tells us a whole lot. We've got this huge micronuclei here. They've got a little mini one just there. We've got these abnormal looking nuclei, not this nice sort of oval shape. Got all these funky little indentations. That's a classic teller of mitotic catastrophe. So these cells aren't having a lot of fun. And then we can score this. So what we did is we treated the cells with doxorubicin, as well as washed it out and let them recover. So maybe we can stop the doxorubicin will cause a cell cycle arrest, give them time to try and deal with that drama in the recovery period, and then we can measure how many aberrations there may be. And what we found is that after that recovery, the cells are accruing more and more issues. So that's kind of all I really want to talk about today on this particular aspect. Not too much in the way of DNA repair. I'm going to try and wrap it up shortly, but we know overexpressing CDCA3 is causing all sorts of dramas for the cells. We want to know, well, really, what does it actually do? Is it only through controlling the levels of that WEE1 that I spoke about on the earlier slide? So we've gone and done a whole bunch of proteomics analysis using some mass spectrometry, which is kind of fun. I don't run the machine, thank goodness, but I'll set the samples up and send them over, as well as BioID. BioID is to help us identify what proteins are hanging out within proximity. So we can put this little tag on the end of CDCA3, which essentially has the ability to add a biotin label to proteins that are nearby. And we'll pull out all those biotinylated proteins, and that tells us what proteins hang out within the vicinity of CDCA3. And we combine that with quantitative proteomics, like what's up, what's down when we overexpress CDCA3. We can come up with some cool maps like that. And generally, we've found that CDCA3 hangs out in programs that control genome instability, would you believe, with topo1 and topo2. To validate that, we've performed some immunoprecipitation analysis, some IPs, using an antibody to pull out one protein, stain for the other. It means that they're hanging out in the same sort of space, or perhaps protein complex. So with CDCA3 immunoprecipitates with topo2. We found that there's some programs that control stress granules. Not going to talk about that today, but essentially that's like a ball of RNA with some RNA binding proteins that they form after stress. So these are transcripts that a cell might like to hang on to help that cell exit the stress state. Cells that are controlling cellular growth, programs that are controlling cellular growth, in this case with mTORC1, which is a really cool, huge protein, in particular, a different protein complex. And here's mTOR. We've tried to work out the region, like I said, it's a massive protein, the region in which CDCA3 hangs out with mTOR. So we've taken a whole bunch of different fragments of mTOR and expressed them in cells. And we couldn't go too fine-tuned as we would have liked, but it hangs out towards the back end of this protein, towards the C-terminal domains of mTOR. But when we do overexpress CDCA3, we see less mTOR localizing to the lysosome, and that's where mTORC1 performs a lot of its functions.
Can you just explain that first one that you did there, the fragments?
The fragments, yes. Yeah, so this is another immunoprecipitation that I was talking about. So we've co-expressed full-length mTOR versus, as well as fragments, shall I say fragments, regions of mTOR. So this heat repeats up to about here is about amino acid 1 through to 1482. Like I said, it's a big boy. We've got this 1271 through to 2008, which is about this region here, and a 1750 to 2579, that's about this region here, as well as a kinase dead. So kinase dead, what we did is we took the catalytic domain of mTOR, we mutated that out so it's not catalytically active, because mTOR is a serine threonine kinase. So what we found here is that when we immunoprecipitated CDCA3, these regions all came down except for 1 to 1403. So we know that CDCA3 is not able to associate with this region of the mTOR protein. Is everyone following along with me there? Is that making a lot of sense? Yep. Cool. So the rest of our research is trying to work out, well, if it's binding up this end, why? And is it controlling mTOR? This data suggests that it perhaps might, but actually I think what we've sort of worked out is that it's controlling its activity indirectly by regulating where it localizes to through a cool protein complex called the gator complex. We don't have many of the gators here because we've got more crocs, right? No, sorry, that's shocking. All right, we're going to finish it up there. And then another cellular program called PLK1. I'm a big fan of PLK1. I'm not going to talk about that today. What I am going to finish on is more of the cool stuff that we've done with clinical samples out of the PA hospital. So we've been able to work with some folks. Rick Thompson, is Rick able to make it this year? Rick.
He's on the lead at the moment.
Yes, yeah. So Rick, a little shout out to Rick who's coming up. He's kicked off this CPAC over here, which has been able to accrue a whole lot of breast cancer samples and of a whole variety of other tumour types as well. We've taken some of the tumours that he's been able to collect, grow these organoids from them. People heard of organoids before? Yep.
They had Charlie.
Charlie, you've had a wonderful introduction to the organoids then. Yeah, yep. So he did some really cool staining of organoids and has done and is doing. I am doing some bog standard immunohistochemistry for CDCA3, but more so because we did it over here, we need to show consistency here for the manuscript. But yeah, Charlie's a real whiz for some of the microscopy staining you can get done. And a lovely chap too. But basically, we've taken our breast cancer samples, stained them for CDCA3, and used the same scoring method that I showed on those earlier slides. People remember those on the TMAs? Nice. Taken these organoids, treated them with, again, increasing concentrations of doxorubicin. Now, I couldn't do as many points on the curve to get that nice sigmoidal curve that I showed last time, partly because it's really hard to grow these things, and so I could only treat them with so many concentrations. But that was enough to tell me that there were varying responses of these organoids to doxorubicin. So again, we performed, instead of an IC50, area under the curve analysis. So the more area, the less sensitive they were to doxorubicin, right? And then we can correlate that AUC value back to the scoring method, so how much CDCA3 is hanging around. And we found that the higher the CDCA3, again, the lower the AUC, so thus more sensitive to doxorubicin. It's all sorts of lovely, nice and consistent. CDCA3 high, more sensitive to chemotherapy. So what I hope I've sort of shown you in this first sort of half an hour is that maybe CDCA3 has some biomarker potential to identify tumours that are responsive to doxorubicin. And we've started to look a little bit more at the function for CDCA3 and know how CDCA3 binds to some of these proteins. And I talked a little bit earlier about how it's forming a secondary structure with binding a protein. This is one of those secondary structures that CDCA3 forms, and that enables it to bind to this protein here. I'm going to finish it there. Thank the folks that have helped with this work and the people that have given me some money to do some of that work. And now it's time for you guys to ask a question. We've already got some hands. Yep.
You go that way.
Yep. Oh, we got some hands? All right. All right, there's one. All right. Who else wants to ask a question? There we go. I don't know. We're not going to do all this. We'll see what we can do. Anyone else? Anyone else want? You got 101.
All right, is that it for the moment? All right, so has anyone got one? Because, we've still got some here.
The number one.
Who is number one?
No.
2, 3.
Oh, yeah.
I was just going to ask about the off targets so it wouldn't disrupt other cell cycle normal. What have you done to sort of avoid that?
Yeah, so targeting CDCA3 itself. Yeah, so we can't target CDCA3 itself just yet. What we're doing is we're trying to mimic CDCA3 because we remember how I said CDCA3 high is doing better. But we know that CDCA3 plays a key role in the mitotic module. So we do actually want to influence that module negatively to increase the sensitivity to chemo. But when it comes to developing an agent, yes, that's really important consideration. We want to only be CDCA3, if that makes sense.
So part of Mark's problem with that is you saw the alpha fold model. There's not a lot of information about how it actually folds by itself, and therefore where you could perhaps put a drug to interfere with a specific function. But having said that, did you see the model at the end where it's interacting with something else? Well, now that's telling us about, maybe a protein interface that might be important on the other protein as well. So the more knowledge that we have about how proteins form the tertiary structure and how they interact with other things can only help, but it's hard.
It's mighty tough. And because what complicates it even more, I'll give you a hot tip, a little bit of inside word before you buy some shares in the company. I don't have a company. Then there's no shares, so you can't buy anything. Is that it actually interacts, we found that there's 3 key spots that interact with this particular protein. So how do you go and design something that hits those 3 spots? That's the challenge we've got at the moment. But yes. Well, that alone isn't enough to have a maximal. We've got it. Yeah. Oh yeah.
All right. Sorry.
Anyway, number 4.
Okay.
Yo.
Sorry, you actually kind of answered the question I was going right, but I had 2 questions anyway.
Cool.
I mean, first of all, I was just thinking when you were talking about the fact that it was a very floppy structure, and obviously has quite a bit of mobility. That obviously changes when it actually forms that secondary structure. So is it that floppy structure that actually allows it to conform so easily to other proteins?
Yep, that's why they say like drunk people don't get injured, right? Because they just stay floppy, No, don't quote me on that at all. But it is that floppiness that does enable it to bind into some of those regions. We think,
I think the other thing you've got to remember about these models is what we've tried to do is take a protein out of its native environment and then we've crystallized it, and in whatever the reagent is that we've crystallized it in, this is the best we've got in terms of folding. Probably doesn't look like that at all when it's actually in cells.
Yeah.
Natural environment.
Yep. There's lots of challenges, but we'll do our best and But yeah, great question. Thanks for that.
Number 5. No, 6.
Yay.
So when you were choosing candidate proteins to pick in research, was there other
how do you sort of decide on CD?
That's a ripping question. Okay, so the story I've just told you isn't necessarily chronological. But for one, I already had some data on CDCA3, and then in an unbiased approach, CDCA3 was there, and I was like, this is hot. That's amazing. Having said that, there is also lots of other fascinating genes in there that I have been working on. So Aurora B, for example, we've got a lot of data on. PLK1 that I've talked about before, it's in here somewhere. There it is. Boom, right there. TBX2, that controls the kinase, which controls PLK1. So I'm working on a lot of these genes as well. But what we do know is that there is a lot of clinical trialed drugs targeting some of these druggable targets, and they don't do squat. So what that tells us is that it's more than just taking out one. We've got to take out a few. But there are some cool transcription factors in this green module, which we are also following up on. So yes, but great question. Now,
7?
we've had 6, right? Have we?
Yeah, Nicole?
You mentioned at one point that the CDCA3 expression is highest in the triple negative breast cancer. So kind of a two-part question was, one, is there any idea as to why it's higher in that form of cancer? And two, because it's highest with that, are you focusing more on targeting triple negative for now?
Well, so with the first part to that question, yes, we do know a little bit about CDCA3 gene, is that it is partially hormone regulated. So it has one part of a hormone response element within the promoter region, but not the full, which we sort of suspect is regulating its expression at the transcript level. At the protein level, it's a difficult one. We know that the protein itself is regulated by a ubiquitin ligase complex, so that's degrading essentially CDCA3. And what we think in the triple negatives is that perhaps that the protein is more stable in the triple negative breast cancers versus the hormone positive breast cancers. Again, we still don't know completely why. But the other interesting thing is that when we go back to the clinical trial data, is that we know in those tumours that have the androgen receptor activity, those tumours have high levels of CDCA3. So we think there's a lot of complex, we don't know why, between the ER/PR and the AR hormone receptors. So we're not quite sure on that front. Now, when it comes to triple negatives, yes, that'd be wonderful. But I think that there's a lot to do in the other breast cancers as well, which are more prominently diagnosed and are greater burden in the clinic when we talk to clinicians because they see them every single day, these patients. But we do know that the levels are low in those tumours. So if we can increase CDCA3 function in those tumours, it'd be great. Yep, thank you. 8, yeah, 8. Yeah, oh yes, you did take a question. All right.
Yes. I was wondering, in terms of, so similar kind of point, the CDCA3.
That's all right, I have problems spelling it too.
You said it's more responsive to chemotherapy and that the expression level is prognostic. So it's quite a similar question. Is it more prognostic the higher the expression levels, and that's why it's more responsive to chemotherapy, or is it prognostic as in, like, how does that work?
Yeah, so essentially we've been kind of all we can do is what we can do when it comes to a pathologist looking down the microscope at the tissue, and so we've done our best to categorise on those 4 expression levels, none through to high. If we were to dig a little bit deeper, we may be able to discern greater levels of prognostication, shall we say, but I think we would lose a lot of power in doing that because the numbers of tumours would be quite low on that front. So we've stuck with the staining and the scoring method that we've got, one part because we see some statistical significance. But yeah, sure, there's more work to do there.
Okay, great. Thank you.
Yeah, cool. Thanks.
None. Joe.
I know we've kind of heard of biomarkers like ER and HER2, which you mentioned. I was kind of wondering how, well, those, we use IHC to detect them, from my understanding. Is it the same for CDCA3?
Yep. So that's what I've showed you before, is that the immunohistochemistry we've done could perhaps be applicable to a tumour. That's what we do have. We've got our own antibody which stains the tissues, and that's what we've used to discern levels of CDCA3 in the tumour. That, I think it'd be wonderful. All the workflow is already there in a pathology lab in a hospital to do immunohistochemistry. There are lots more exciting approaches like digital pathology that are coming through. So who knows ultimately? But at this very point in time, it's a great discovery tool to tell us that CDCA3 may be a biomarker. Yeah, cool, thanks.
You obviously did it with your example in breast cancer. Were there any other cancers that possibly had the CDCA3 expression?
Yeah, great question. Yep, yeah, and so the answer is yes. Yeah, so lung is what we've done and published already, and yes, prognostic. We have looked at colon cancer. It is a little bit strange in that much of what I've shared already is the complete opposite in colon cancer. Got a student at the moment having a look at pancreatic cancer. That's interesting, where again, it's much like it appears in the tissue microarray data that high CDCA3 has a better outcome. So there might be something going on in the gastrointestinal sort of regions. We don't know what's going on there. But yep, thanks for your question.
Have you found any other drugs you think are worth investigating other than the ones that you're currently working on?
Is reference to doxorubicin? Yeah, so the reason I use doxorubicin, and I probably should have said it as well, is that's a standard of care chemotherapeutic agent. So we really want to just use that because it's used in the clinic. Any other drugs? Like I said, we spoke about cisplatin as well, so it's another DNA damaging agent. We've looked at other DNA damaging agents which are used across those other cancers that from the previous question. So we're just looking at standard of care initially. So yeah, we would love to then go and make our own, but we're still a fair way off really testing those out.
Have you found any adverse effects from that drug?
From doxorubicin? Yeah, so ladies that are treated with doxorubicin have a lot of side effects, and this is where I'm working with consumers, some ladies that live with disease that tell us a little bit more about what it's like to be treated with some of these agents, and just quietly, it's not awesome. So that needs to be weighed up against the benefit, as well as strategies to mitigate some of those side effects. So yeah, there are a lot of side effects with chemotherapy, but yeah, thanks for your question. Good to be aware of. And that also does drive some of the work too. If we are able to identify tumours that might require less treatment, so if we can reduce over-treatment, that's nothing but great for the patients. Yeah, good question.
If you essentially target the CDCA3 protein, do you think that there would be any or mechanism of cancer?
Yeah, great question. I'm willing to say yes, because we do know how cheeky tumours are. So that is absolutely something to consider. Yeah, so that's something to almost worry about when we get there. But yeah, good thing to test and to try and model.
Is there a reason why you chose deoxyribonucleic over cisplatin in the final test?
Chose, sorry? Oh, doxorubicin. Yeah. Yeah, so that was mostly, again, because doxorubicin is the standard of care. Cisplatin is used, but it's still going through a variety of clinical testing, yeah. And when it comes to oncologists, there is a bit of dealer's choice, shall we say, when it comes to the different chemotherapeutic cocktail that they may use. So, but doxorubicin is the core of that particular treatment cocktail, yeah.
I need to take my shoes off to go any further. So
Self-regulate.
18. So I think a lot of my questions about like the presentation itself have been answered, but I was wondering in terms of if you can get this kind of published, what would then sort of be your next steps if you're allowed to talk about that?
Yeah, publish more. That's
So just do more tests to show
Yeah. So, I mean, for us at the moment, when it comes to breast cancer, we won't be doing any more work on this until we get more funding. But the other focuses are on other cancer types. But essentially, what we would love to do is to apply that whole module work that I showed early up in the talk to these other cancers and to do a direct comparison to understand the reliance on specific modules for certain solid cancers. Yeah, that'd be cool. 19? 18?
18?
Oh, no.
You were 18?
Oh, 19.
Okay, I think 19 is next. Thanks, Carl.
I was just wondering, are there any sort of like mutations in the genome that you attribute to the levels of the CDCA3?
Yeah, it's great. Ripping question. So CDCA3 itself is rarely mutated. There are a couple of mutations here and there, but not within any of the structured regions that we're aware of in those floppy regions. So what on earth they do, we don't know yet. We have found that the CDCA3 does tend to correlate with, in terms of lung cancer, mutations within EGFR, the epidermal growth factor receptor. There is mutations within KRAS, CDCA3 higher than KRAS, as well as another mutation within LQB1, which is also called STK11, and that particular set of genes control AMPK, which is a stress signaling transduction pathway. So there are a couple of different mutations that levels of CDCA3 associate with.
Okay.
Yeah, thanks for that.
Right, 23. This is probably the next one.
Oh.
Gets tough towards the end, but
Oh yeah, what's left over?
I'm just wondering, just because you obviously started with lung cancer, what correlations you've kind of found between lung and breast cancer?
Yeah.
Obviously, they both have the CDCA3.
Yep. Yep.
I've seen in my DNA PRCC1 also related to breast cancer as well.
Yes. Yeah. Great question. So, so they're both epithelial origin, tumors of epithelial origin, and CDCA3. Sorry, lung and breast are very similar when it comes to CDCA3 biology. We know actually. Oh, this is some stuff I didn't present that I will show in terms of DNA damage repair. This HR, homologous recombination, is a DNA repair pathway for those DNA double-strand breaks. And the D stands for deficiency, so tumors that can't do that because of a whole raft of reasons. We see in lung and in breast that the levels of CDCA3 correlate with this deficiency in homologous recombination repair. So high CDCA3, not much chop at repairing through the HR pathway, so they're reliant on the other more error-prone pathways. So that's why we're seeing a lot of these chromosomal aberrations that we see on the right. So we can see that some of these tumours have lost whole chunks of chromosomes or duplicated whole chunks of chromosomes. Remember I showed you about the micronuclei before? Some of that is indication that there is large, whole-scale, just chop outs and unable to deal with the genomic data or information. So they just chunk it into this micronuclei. So that is a whole bunch of DNA repair that we see consistent between the lung and the breast. So yeah, that's a cool question.
Based on the bio-ID connectivity to CDCA3, assuming that you can completely rebalance the expression of it within the cell, are there any predictable, known, or expected potential adverse effects in those connected systems? Might dissuade the use of it being a target?
Interesting question. I'm certain there would be. Nothing's perfect for one, and that's what we've done there is in the proteomics is to increase the levels of CDCA3 and to look at what proteins are deregulated. A lot of those proteins we do find consistent between the protein data and that module transcript data, so RNA and protein. So there are consistencies there. So we're deregulating and destabilizing that module. When it comes to any issues with doing that, we don't know yet. So
you said that colon cancer is the opposite. So if you CDC.
Yeah, sure.
The expression in one area will lower
Yep.
the chance of increase the risks in other developments.
Oh, you're right. I see. I understand what you mean. Yeah. Look, I get that's a long-term sort of study, and we really have no idea just yet. But I think it would depend as if it is to become a treatment, which is again also a long way off. We would need to know the tumour that we're treating and the disease state a whole lot more effectively. So yeah, look, that's a good question, but tough one to really answer at this point in time.
So Mark's going to answer the next 4 questions fairly quickly.
Oh yes. Oh yeah.
27.
I was wondering, after getting you said that there's less secondary part of the study.
Yep. Well, they're classically undruggable because they don't have a nice structure to develop a drug to poke into that little pocket. So yeah, that's what makes them hard.
Did you do the same thing that I did?
I didn't, but maybe I just sort of tried to fold my arms. These are my gang moves. Sorry, I'm totally cool. All right.
Of the tertiary structures that you found with CDCA3, you said that there was like 3 little arcs. Are you trying in your potential drug to target all 3 of those?
Well, we've looked at hitting individual spots by taking a peptide that mimics that, right? And mutating at the spot where it binds in those 3. And that peptide is only effective when it hits all 3. No, so it's 20 amino acids.
Molecular weight?
I couldn't tell you off the top of my head. Yeah. So we're looking at developing like some linkers between small molecules, which would reduce its size, but it's not easy. Yeah. Yeah. There you go.
32.
Yes. So you showed there was the mTOR gene.
Oh yeah, protein. Yeah.
There was the heat region. Why is there a heat region? What does it do?
Yeah, that's a really good question. So that heat domain. It's just really hot, right? No. I'll have to get back to you on that one. The heat domain, I think, plays a role in binding other factors in other
Hang on,
proteins.
we've got one more question.
Yeah, sorry.
Oh,
we got a couple.
Yeah, Tom. No, it's Tom.
Yep.
I'm 35.
35. Let's get Tom.
So before, when you were investigating anthroposis in CDCA3 other genotype linkages, what one would be more likely to further investigate?
In terms of that module, I sort of referred to transcription factors before. So the transcription factors that underpin the expression of a lot of those factors, if we can take them out of action, there's every chance you're going to take out that entire module or chunks of that module. So that's what I'm keen on looking at. Thank you.
Sorry, no, that's okay. Mine's just a quick question. You looked at breast cancers. Was any of them male? Like from male breast cancer?
No.
Like I know it's rare.
Yeah. Good. It's a really fair question. Yeah. No, they weren't. Yeah. Thanks for that.
Given this hormone regulation thing and the suggestion that AR might be involved, because of course that would be stimulated by the testosterone in male and sexual rather than female.
Yep.
Interesting.
Yep.
All right, let's thank Mark, and I'll come to you for information later.
All right. Nice. Thanks everyone. I'll stop this.
Yes, obviously stuff.